So now that we've looked at the overviews of photosynthesis and cell respiration, we'll look at the process that photosynthesis uses to harness light energy. In other words, we're really focusing in now on the light dependent reactions. And so who can do this? It is done by what are called photoautotrophs, photo meaning light, auto meaning self, and troph meaning the kind of level that an organism is on in terms of the food chain. So in other words, light self-eaters really just means that these organisms are capable of making their own organic molecules and they don't need to eat others. They can just rely on themselves to be able to do this so long as sunlight is available. And so because all other organisms on Earth are going to rely on the organic molecules that they produce, they're also going to be referred to as producers. And these are going to be your plants and algae, which can both do photosynthesis as we're going to be looking at it now, as well as cyanobacteria, which are share a common ancestor with modern chloroplasts, but they do not do photosynthesis quite the same, but that's okay. Okay, so the majority of oxygen on Earth is actually produced by algae, but because we're much more familiar with plants that are on land, let's take a second and look at the structure of those plants because it's important to note the relationship between the structure and the function of the plants. So for starters, the photosynthetic organs that plants have are the leaves. And they're very, very flat, and that's going to allow for a maximum amount of surface area to volume. This means that it's just, maybe 100 cells thick, and material can diffuse easily either from the environment or from the cells and out the leaves and into the environment. And to facilitate this process even further, we're going to see these little guys here. These are called stomata, and each stomata has a couple of guard cells that, when they tighten, open up to make these little pores. So the stomata itself is the opening as well as the cells that surround them. And that's going to allow for the exchange of gases. So bringing in carbon dioxide during the day and releasing oxygen during the day, or flipping that at night. We can see here a sample of a cell, and the most obvious organelle throughout this entire thing are all of these chloroplasts. Right here, we've got our diagram of the chloroplast, and I apologize, I don't have my iPad queued up and ready to go, so you're going to have to review as we go along what the different structures are. So in the chloroplast, we've got an outer membrane, an inner membrane, and we've also got the thylakoid membrane. So if you see here, all of these little folded bits are called thylakoid membrane. Each of these little coin-looking things is a thylakoid, and they form stacks called granum, plural grana, singular granum. And essentially, what used to be the prokaryote's cytoplasm within this chloroplast is now referred to as the stroma. Within each of the thylakoids, there's a space, which you may hear referred to as either the thylakoid space or the lumen. I think in the PowerPoint coming up, I mainly just do lumen because of space. Like I don't have enough room to type it out, but just be familiar with both of those terms. Okay. So let's look at what is light. Light is really just energy that travels as waves, and it's part of the electromagnetic spectrum. So any energy that travels as waves is electromagnetic energy. Our eyes have certain pigments, certain molecules that can pick up the particular wavelengths of light, and we can recognize those as visible light. Everything else is too damaging to our eyes or too low of energy to be able to excite our neurons to be able to recognize it as visible. And so this is where we've got the Roy G. Biv, and as it turns out, plants also have these pigments, just like we have pigments in the back of our eyes that pick up the different wavelengths of light, so do plants. The primary one that you'll want to know is chlorophyll. There's actually two forms, chlorophyll A and chlorophyll B. There's also carotenoids, xanthophylls, and a number of others that when the chlorophyll stops being produced as the temperatures drop, then we can start to see a lot of those other accessory pigments. And so what they're going to do is they're going to absorb a very specific wavelength of light. And when that light strikes the pigment, it excites the electrons. And so if there's enough excitation, the electrons can actually leave the pigment molecule. But for most pigments, that is not the case. And instead, what we have are these big complexes called photosystems. And there are two photosystems that we're going to be looking at in the plants for the light-dependent reactions. So here's a photosystem, and this has a ton of little chlorophyll and other accessory pigments that are all picking up certain wavelengths of light. And it's going to channel a lot of that energy rather than having a whole bunch of electrons that get excited but then fall back down to their ground state. They get a little bit excited but then pass that energy on until there's enough energy funneled to one single molecule. And that is called the reaction center molecule. And that molecule is going to be able to give up a pair of electrons. And that's where the fun really begins. So in terms of photosynthesis, we're going to be looking at photosystem 1 and 2, but we can't make things easy because, well, we don't discover things in logical order sometimes. In terms of pigments, each pigment is going to absorb, as I said, very specific wavelengths. So the chlorophylls tend to absorb the blue and a little bit of red. What happens to the green and the yellow is that they're reflected. So they hit the pigment, bounce off, and that green or yellow light then is capable of hitting our eyes, which is absorbed by a pigment in our retina, which goes, green, or yellow, or fire at the same time and gives you the appropriate shade somewhere therein. For the carotenoids, they're doing a lot more. There you go. They're absorbing a lot at the blue, kind of into the green wavelengths, but nothing in the orange or reds or yellows, which means those lights can bounce off and be interpreted as particular colors to our eyes. So the absorption spectrum is specific to each of the different pigment molecules, and the pigments that are found in a particular plant collectively will make what is called an action spectrum. So this is all the different wavelengths that the particular photosystem or the particular plant is capable of utilizing or harnessing. So again, here is our chloroplast. Just to re-familiarize yourself with it, actually pause for a second and see if you can name everything here. So we've got did you pause? I hope so. Outer membrane and inner membrane. Each one of these here is a thylakoid membrane that make up a thylakoid. Within those thylakoids is a liquid, like a cytosol type liquid, but we're going to refer to those as the lumen or the thylakoid space, and all the space around them are the stroma. One stack of these little guys is a thylakoid membrane. And if we zoom one stack is a granum. I was getting ahead of myself. If we zoom way into the thylakoid, we see that, yeah, this is the same type of membrane that we've been seeing. And in fact, there's a whole bunch of proteins that are embedded in this thylakoid membrane, just like what we saw in the mitochondrial inner membrane. So let's look at the process of electron transport. While I get myself comfortable. Okay, so a couple of things that we should do first to orient yourself. So again, we're looking at a thylakoid membrane. These are the different proteins that are embedded. And in the electron transport chain of cellular respiration, I did not expect you, I don't want you to memorize the names of each of the pieces, each of the components that are here. However, I do want you to know 4 parts relating to the electron transport chain, and I will make sure to have that in the comments below because I did not label them on here. But we've got photosystem and another photosystem, and each of them has a reaction center with a pair of electrons that can be donated. And so as it turns out, this is photosystem 2. This is photosystem 1. You're welcome. And then in between, there's just a bunch of other proteins that are involved in moving things along. Interestingly, one of them, you don't need to remember this, is called ferredoxin, and ferrous because of the fact that it has iron. So really, really green plants like spinach give you a lot of iron because of all of that ferredoxin. Anyway, fun fact. And then, so we've got the 2 photosystems that are involved. We've also got this guy here is an enzyme called NADP plus reductase or NADPH reductase, depending on what literature you're looking at. As you can imagine, that's going to be responsible for the reduction of an electron carrier to form NADPH. And of course, we've already met this guy here, ATP synthase. All right, so we're going to start with light striking the first, or striking photosystem 2, the first one in the chain of events that we're going to be looking at. Make sure it's blue or red light. And then that's going to be able to excite the pair of electrons. Once those electrons have been excited, they're going to leave from photosystem 2 and move through the electron chain into photosystem 1. As they're moving through the electron transport chain, just like we saw in cellular respiration, we're going to see the pumping of protons into the lumen. So you kind of see that process happen there. Now, as it turns out, photosystem 2 is ridiculously electronegative. And now that it's lacking a pair of electrons, it's got to get them from somewhere. And so photosystem 2 actually is more electronegative than oxygen, so much so that it can rip a water molecule apart and leave it with nothing but a couple of hydrogen ions and oxygen. And so this water gets broken down, and that's where the plant is producing this oxygen, which is a byproduct of photosynthesis. Okay. Then we have red or blue light striking photosystem 1. So the pair of electrons that was here have gone into photosystem 1. Photosystem 1 is going to be struck by enough light to excite it. And that's going to allow those electrons to escape and move to the NADPH reductase, where we're going to see NADP plus reduced into NADPH. Ta-da! So if we're following the movement of these electrons, we can kind of start with photosystem 1 to make it make more sense. Light strikes photosystem 1. This pair of electrons goes into NADP plus to make NADPH. To replenish this pair of electrons, it's going to require that an electron pair is lost from photosystem 2, which will get excited enough when enough energy is absorbed to move it through the electron transport chain and replenish the electrons that were lost in photosystem 1. And then finally, when photosystem 2 is short on electrons, it's going to rip apart a water molecule in order to get the electrons that it needs. Okay, so all of that should look pretty familiar, but also way different than what we saw in cell respiration. It would behoove you to take a moment and compare and contrast the 2 electron transport chains between photosynthesis and cell respiration. Really think about what is your source of energy that fuels that powers up the electrons enough to move through this system. Is it a pair of high energy electrons that came from another chemical, or did they have to get energized from some other source? What is the ultimate source of the electrons in both cases, and what is the ultimate ending spot for each of the electrons? So think about that. And then we can look at chemiosmosis. And no surprise here, it's a ton easier than the electron transport chain. all that happens is we're going to take this electrochemical gradient that is built up because of the electron transport chain. And so now we've got a ton of protons that are chilling in the lumen, not so many in the stroma. Think about something here. We talked in the beginning of the year about water that dissociates and makes hydrogen ions and hydroxide ions. So what would you expect the pH of the inside of the lumen to be as it compares to the stroma? Another kind of side conversation that you should have with yourself, or maybe talk to your group about it and see if you can figure that out. Anyway, just like what we saw in chemiosmosis during cellular respiration, we're going to see that the protons are going to flow through the ATP synthase from an area of high to low concentration. And as this occurs, the ATP synthase will spin, giving enough mechanical energy to smack ADP and a phosphate group into ATP and form your ATP, okay? Now note that you don't need to remember how many of anything are used during photosynthesis other than the overall equation. So far, one molecule of, where do I want to go with this? Okay, so far we need 6 H2O, and that is going to be involved in the formation of 3 oxygen molecules, just for this part right here. And then we'll add some more on later on during the second step. Oh yeah, I said that already. Apparently I should have pressed a button. Okay, now we're done.